Hello everyone, welcome to the paper discussion of biochemistry and analytical techniques. Over here you will find that there are three sections in this paper. Section A, which has to be done in the form of 5 questions, 1 mark each. Section B has 5 questions, 4 marks each, and section C has 5 questions, that is 7 marks each. So for section A you have 10 questions, that means you have a choice, and out of them you have to select any 5. So the ones that I've ticked, they are the ones that I've chosen. Okay, now for section B, can you see there are questions such as explain in brief ribonuclease A. So you will not find these enzymes directly in the syllabus. I'm going to discuss the syllabus also with you in just few minutes, but you can see you have to make a choice. Like if you want to write a 4 markers, you have to be very specific for the questions that you're selecting, because you have to write more content. That means you get a sheet of 32 pages and you have to fill that sheet. So you should be having a content to fill at least the main sheet. Apart from that, you can take an extra sheet which is of 10 pager, but just make sure the questions that you're choosing, it should at least give you the content to write 4 pages for each question. Okay, in the first part of section B, I chose the classification of enzymes. That was easy and we have already made a video on it. Explain in brief DNA structure and its types. So all the helicase types which are there, A, B, C, alpha, beta, gamma, which were there. So that could be discussed over here. And in brief, DNA structure. So make sure if such questions is there, you have to make a structure of the DNA also, showing that how the nitrogenous base is linked to the sugar and how a phosphate group has been attached. That basic structure should come in your answers. Wobbles hypothesis. Though I knew the wobbles hypothesis, but since I thought I have more content on post translation modifications in eukaryotes, so I chose the second one. Rather, this was also a very direct and good questions. Over here in this paper, there are two things that you have to know: wobbles hypothesis, Ramchandran's map, and one more. It's not an hypothesis, but it's a concept, that is Jablonski's diagram. So these three things you should practice properly, because every year out of these three, at least one or two is asked. Okay, so there's a question of chromatography, and very specifically they are asking about thin layer chromatography. So one question you'll find that in general there's a question on chromatography, and over here that's a specific question asking you about the principles and applications. So be very specific while you're answering such questions. And the one that I chose was mechanical method of cell disruption. Over here you can include the centrifugation that is there, and you can explain the complete process, so it's easier that way. Okay, and then the next question 5 had basic principles of light microscope and its applications. I didn't wrote it because I didn't had much content on this, because light microscope में थोड़ा सा ही है, and उस microscope का diagram बनाना it would have been very difficult and time taking. So I chose spectroscopy and Jablonski's diagram because it was very direct and had a lot of content on this. Now in section C, you can see there are 5 questions that are supposed to be attempted, and each question is of 7 marks. That means at least 10 pages, yeah, maybe 10 pages. That means 5 pages front and back should be written. So the first question was on protein classification functions. Very easy. We have discussed about protein in the video, and classification very easy based on functions, and like based on structure, like simple protein, conjugate protein, and like you can write whatever criteria you have studied, you can include all of them. I just generally included function and structure one. This equation, I don't know, I didn't study this, so I had to skip it, no point. DNA repair mechanism, I knew the diagram, but contents was very less. For 7 marks, no point writing this. Better option was gene targeting and gene disruption methods. Gene targeting, at least you can write 3 pages. Gene disruption method, 3 pages for it. Matlab, lot of content for this. Translation in prokaryotes and post transcriptional modification. So I don't know why have I not ticked it, but I think I wrote about the post transcriptional modifications, though I didn't had much content to this, but I just thought कि maybe both of these questions are of similar nature, दोनों में उतना ही है, तो at least 2-3 pages were covered. Over here again, there's an option on centrifugation principles and ion exchange chromatography. Though ion exchange chromatography is very easy, but mostly we cover up with the definition and thoda at least one page is only there, and application 1 or 2 page. So hardly 3 pages could have been covered. On contrary, centrifugation principles and types. So there are many types of centrifugation and principles के अंदर भी RCF factor and lot of things can be included. So I chose this one, not this one. And over here already I have written content on spectroscopy in the previous answer, so that was there freshly in my mind. I chose the UV visible spectroscopy and its application. Whereas difference between TEM and SEM, these difference, there are hardly few difference between TEM and SEM, तो 3 पेज ही कवर अप होता है, तो उससे कंपैरिजन में मुझे स्पेक्ट्रोस्कोपी ज़्यादा ईजी लगा. So now this differs on person to person what you want to choose. Both of them are doable. So the paper is pretty direct, easy, but it's just you need to be well versed with the content. Okay, now coming to the syllabus of paper 1. So this is biochemistry and analytical techniques. So over here, these are the important things that came up in the exam and I've highlighted them with orange. You can pause this video, take a screenshot of this and revise these topics nicely, because this time the questions were not very direct, but yeah, thoda sa they are shifting towards analytical questions. मतलब एकदम shift तो नहीं आएगा, but हाँ, they are trying to put focus on more conceptual part of the syllabus. Like not just like earlier they used to ask about Ramchandran maps, protein pair, enzymology पर ही major focus था, but this time they have shifted to nucleic acids and transcriptional changes. तो थोड़ा सा application oriented part को उन्होंने ज़्यादा पूछा है. Okay, and this is the second part. Over here also, you have just highlighted the important things that have been asked. Around this, you have to work and prepare for maybe next year, those who are going to attempt this.